So hello and welcome to Bioaffairs. Today I'll be talking about gram-negative cell wall. Whatever you are seeing in this screen is the gram-negative cellular structure in 3D form. Now I have marked the cell wall structure so that you can understand specifically where it differs from the gram-positive bacteria. Now let us understand this outer membrane structure. So the unique characteristics of the gram-negative bacteria is the presence of outer membrane. Now this outer membrane is absent in case of gram-positive bacteria. So what is the composition of this outer membrane? Why it is unique? So outer membrane is composed of porine proteins, as you can see here, porine proteins. The braunslipoproteins present in the gram-negative outer membrane and several other integral proteins and LPS layer. So LPS layer is the most significant aspect of outer membrane, though braunslipoprotein is not behind also. This outer membrane is attached to peptidoglycan layer. These capsular structures, whatever you're seeing here in the yellow and the brown marks, are nothing but the NAG and NAM molecules. So this thin structure, these thin structures going along is the peptidoglycan layer. I have marked it. Now this peptidoglycan layer is attached to the outer membrane. This outer membrane, how? Now the braunslipoprotein, these structures are the brown lipoproteins which adhere to this peptidoglycan structure. Means they are making the link between the peptidoglycan layer and the outer membrane. So this is why Brown's lipoprotein is very important. Now the unique composition, one of the unique composition of outer membrane is the LPS layer. This LPS layer is composed of three components, that is lipid A, which is not visible here because the lipid A core polysaccharide are the embedded parts of the outer membrane, which are embedded inside this bed, okay, in this outer membrane bed. That is why it is not visible. I have not written here. But the O side chains, these thin hair-like appendages you are seeing here, are the O side chains, which are just out from the or the part of the LPS layer, and the lipid A and the core polysaccharide is behind them, embedded in the membrane. So these are the unique few compositions: the porine molecules, integral proteins, Brown's lipoprotein, and the LPS, which is again composed of three components: lipid A, core polysaccharide, and O specific side chains. So I think it is clear what is outer membrane and how it is connected to peptidoglycan layer. Now, in case of gram-negative cell wall, whatever you are using the term cell wall. So what is the cell wall? Which one will be the cell wall? The peptidoglycan layer in case of gram-positive, or the outer membrane? Here we will call the cell wall. No. So the solution is the composition all total peptidoglycan plus outer membrane is the cell wall of gram-negative bacteria. So whenever you are asked for answering the gram-negative cell wall, you will be writing peptidoglycan layer plus outer membrane. Okay. Now this is the space between the peptidoglycan and the cell membrane. As you can see, this is the cell membrane, this whole structure, and this is the phospholipid bilayers present in both the cases. You can see this is the outer membrane, and in case of cell membrane, the space between the cell membrane and the peptidoglycan layer is the periplasmic space. So there are several enzymes are present. I am not going deep into there because I am not taking that class. So gram-negative cell wall, this is the typical structure. I hope it is little bit clear. Now let's see the specific definitions. What are the actual compositions present inside the LPS layer, or the individual component of the LPS layer? So let's recap once. This is the cell wall of gram-negative bacteria, which is composed of outer membrane and peptidoglycan layer. The outer membrane lies outside the thin peptidoglycan layer. Already I have told you, thin peptidoglycan layer. And the braunslipoprotein, which is very essential in making the direct contact between the peptidoglycan and the outer membrane. It is the, actually you can say it is the cement between the two bricks. Okay, and the bricks are nothing but the outer membrane and the peptidoglycan layer. Now this lipopolysaccharide is composed of three components. Lipid A, I told you 2 times already, I think. Now this lipid A contains 2 glucosamine sugar derivatives. Okay, these are the compositional difference, with fatty acids 1 and the phosphate attached there. So 2 glucosamine sugar with fatty acid and phosphate attached. The fatty acid of lipid A are embedded in the outer membrane, while the remainder of the LPS molecule projects from the surface. That is the O side chains I have already shown you. You can read back the video and you can see it. The remainder part of the LPS molecule projects from the surface is nothing but the hair-like appendages, which is the O side chain. Now again, in between that, the core polysaccharide is joined to lipid A and is constructed of 10 sugars. Now remember, these 10 sugars may differ from one bacteria to another bacteria. Okay, this gives the antigenic variety to the individual pathogen or the individual gram-negative bacteria. And the O side chains, which is the hair-like extending or extruding format, is also composed of peculiar sugars. Means the compositions are peculiar and it differs from one gram-negative bacteria to another gram-negative bacteria, which give them unique characteristic and the capacity to show unique immunogenicity or antigenicity from one individual to the another individual. That is the thing present in the gram-negative cell wall. Now at last I will show you the gram-positive cell wall structure, 3D structure, so that you can easily memorize and visualize the actual difference between both the bacteria in one peak. Okay, so here is the presentation of gram-positive cell wall, a typical bacteria, and you see the cylindrical structure, so nothing but the teichoic acid, which is extensively present in case of gram-positive bacteria, and these capsular structures, which I have already shown in the previous slide, that is gram-negative slide. These are NAG and NAM molecules, totally interlinked, and you can see an aspect, this peptidoglycan layer is much more thicker in the visualization of this image, also much more thicker compared to the gram-negative peptidoglycan layer. So this thickness itself describes its characteristics, but there is absence of any kind of outer membrane or any kind of LPS or any kind of braunslipoprotein. Nothing is present there, but the periplasmic space is there, because the peptidoglycan layer, which is here in case of gram-negative, gram-positive bacteria, is the cell wall, and this is the cell membrane, which contains against peripheral proteins, peripheral proteins, integral. This is the integral proteins. These are the peripheral proteins. Okay, so these are the phospholipid molecules present in the membrane. So this is the cell membrane structure. So you can easily visualize. If you can visualize these two peaks in your mind, you will be easily differentiating these two bacteria. Just remember the crucial points. So thank you very much. If you like my video, do subscribe, and if you have any question, definitely you can put it in the comment section. I will answer. Thank you very much.
If my classes are really helping you in your studies and it is really clearing your concept, give a big thumbs up and don't forget to subscribe my channel Bioaffairs.